I will stop recording before the Q&A session, just so everyone feels comfortable posting their questions. To that point, you can put your questions in the chat, and then we will address them at the end, or at the end, you can raise your hand using the Zoom function and turn on your microphone and ask a question. But we want you to ask a lot of questions. So even if you type it in the chat throughout the talk, as things come to you, that's totally fine, but we'll address them all at the end. And lastly, we just want to keep all discussion and everything respectful. CAP does have a code of conduct that we want to follow. So with that, we will get started. So our speaker today is Dr. Avery Berman. He is at Carleton University, and he's going to be talking to us a little bit about his research, which is in the field of medical physics. So I'll pass it to you,
Oh,
Avery.
okay, great. You hear me okay? See my screen okay still? Thumbs up? Okay, great. Yeah, thanks so much for the invitation, Megan, and for the introduction for the talk. Yeah, so today the title of my talk is Exploiting the Magnetic Properties of Blood to Map Brain Function with Magnetic Resonance Imaging, or MRI. So kind of a mouthful of a topic, or a title at least, but hopefully you'll find that there's some nice lighthearted elements throughout the talk, giving you a bit of an introductory overview of how physics plays a role in medical imaging, and specifically my area of research, which is MRI and functional MRI. All right. All right, so let's start off with why MRI has been so foundational to the field of medicine. And it's really because it's able to produce such a wide range of contrast types that have been invaluable in medical imaging and research. So you can see here different features of brain anatomy that are highlighted by such a wide range of different contrast types. And none of this requires contrast agent injection. It just depends on how we acquire our image. So we can image anatomy. We can also image the connectivity of what brain areas are connected based on sensitizing our MRI signal to the diffusion of water molecules throughout the brain, which follow fiber bundles that connect different brain areas. And we can also measure the functional activity or the brain activity across the brain. This is the field of functional MRI. It's my main area of research. So as an outline for today's talk, I'll give an introduction to functional MRI, both the physics and the physiology that contribute to the functional MRI or fMRI signal. Then give a bit of an overview of my own research, which is more focused on physiological MRI. So understanding kind of function of different tissues throughout the brain, looking primarily at oxygen metabolism or brain energy, and some vascular dynamics. And then I'll talk a little bit on my career pathway and career options within the field, the broader field of medical physics as well. Okay, so let's start with the introduction to functional MRI. So backing way up here, magnetic resonance imaging is based on the phenomenon of nuclear magnetic resonance, or also known as NMR. So the nuclei part is because we're deriving most of our signal or all of our signal from atomic nuclei, which is usually coming from the hydrogen nucleus, which is just a proton in water, which constitutes the bulk of molecules in our body. We're exploiting some magnetic properties of the hydrogen nucleus, and that is that each proton has some spin magnetic moment that we're able to be sensitive to changes in the magnetic moment or the total magnetic moment within an imaging volume. And then finally, we're using a resonance phenomenon, which is a dependence on a specific frequency. In this case, we see a couple different emergences of this resonance phenomenon. One key one is that the spin magnetic moments, when they're in an external magnetic field, we call it B0, they'll tend to precess around the axis of that magnetic field. And that frequency of precession is directly proportional to the magnitude of the main magnetic field. So we call that frequency F, or sometimes omega naught if we're talking angular frequencies. So you'll see omega naught come up sometimes. Now, MRI is the combination of NMR, nuclear magnetic resonance, now with some spatial encoding, so we actually know where our signal's coming from. So here's a cutaway of a standard MRI scanner, and I'll just step us through the different components that are used to generate our MRI signal. So first off, we have a strong magnetic field. It's effectively a solenoid magnet, not like this sort of solenoid, which you'd see in your electronics components, but it's a superconducting electromagnet. So there's zero resistance in the wires and they're cooled using liquid helium in order to maintain that superconductivity. And this allows us to get these ultra high magnetic fields that are necessary to give us reasonable measurable signals. Next, we don't just want to magnetize our sample, but you also need to perturb it away from its new equilibrium. And we do this using radio frequency pulses to both transmit some sort of energy into our system to perturb it away from equilibrium and also to receive our signal. We can use the same transmitter and receiver coil to both transmit and receive. And it's basically just radio technology. It's just basically a coil, a conducting loop that we can use Maxwell's principles to both emit and receive changing magnetic fields. And then finally, in terms of spatial encoding, we use what we call gradient coils to spatially encode. So in this case, we have what's called a Maxwell pair, where we have current flowing in opposite directions in these conducting loops. And this produces a spatially varying magnetic field. And then remember I said that the rate of precession is proportional to the magnetic field in that area. This means that spins at different locations will precess at different frequencies, and we can then infer where our signal is coming from. But the crazy part about MRI is that we don't just take in our image like we do with the camera and, pixel 1 corresponds to location X, Y in space. We actually encoded what's called K-space. This is the spatial frequency domain of our signal, which is really just the Fourier transform of our image. So this is what our image looks like when we get it off the scanner. This is the K-space representation, or the spatial frequency representation of our image. We then just apply an inverse Fourier transform, and we can recover then an image, like here, in this case, the brain. So to give you a sense for the kind of magnitude of the magnetic fields that we're dealing with, some scientists at Stanford were decommissioning their MRI scanner, and they decided to put an empty gas cylinder into the bore of the MRI scanner right before they shut it down to just show the sort of forces that we're dealing with in such a strong magnetic field. So I'll just play this video. Oh no. Maybe it's because I have the pen on. Sorry, one sec. There we go. So pretty dramatic. You wouldn't want to be on the other side of that cylinder. I'll play it once more for anyone who might have missed it. So that cylinder has become a lethal projectile. And that's because we're dealing with magnetic fields that are about 100,000 times the Earth's magnetic field. And as that cylinder gets close to the gradient of the magnetic field, going from very low field to high field, it gets sucked into the magnetic fields, to the scanner. So this is actually the number one health concern for MRI or safety concern for MRI is making sure that there's nothing magnetic that's going to be sucked into the bore of the scanner. This is why we get people changed into hospital scrubs generally to get into the MRI. Make sure that they don't have any unsafe metal on them. Most surgical metals and dental metals are MRI safe though, so we don't have to worry typically about those, although we will do a safety check to make sure that there's nothing that shouldn't go into the scanner. So in terms of how we actually generate this range of contrasts, there's two factors really that determine it. The first one are the tissue properties themselves. So we're mostly sensitive to where the water is. So that's the first thing. So we sometimes call that proton density. So here on the left is a proton density or water weighted image where there's more or less water. But we're also very sensitive to the molecular environment. So whether water is tightly bound, to a protein, for example, or if it's loose and floating around in more of a fluid format. And this will change several different MR-related parameters. They're called relaxation times, T1, T2, or T2 star. And we can see just different ranges of contrast depending on if we're going to sensitize our MR images to these three different relaxation times. And then the way we sensitize to those different relaxation times is what we call adjusting our pulse sequence parameters. So this is just the timing and the power at which we're applying our radio frequency pulses, when we choose to encode our image, etc. There's a whole range of knobs that we can tune to change the contrast in our images. Oh, right. So I'd already shown this previous image that with these parameters in hand, we're able to generate quite a staggering array of contrasts. like we saw before, different anatomical details. We can also get a chemical fingerprint from different brain areas. So here, this is MR spectroscopy, measuring different metabolites throughout the brain. And I know I'm biased, I do brain imaging, but MRI is also invaluable in musculoskeletal imaging. So here's an example of knee image where we're able to see potential damage to tendons and cartilage and the bone as well. Also invaluable in cardiac imaging and angiography, highlighting the vasculature throughout the body here. And for these contributions really to medicine, MRI, the kind of early inventors and developers of MRI, Sir Peter Mansfield and Paul Lauterbur, were awarded the Nobel Prize, but not for physics, actually for medicine or physiology, back in 2003. All right, so I'm just going to kind of break here for a second, give myself a pause, and this is just a fun little interactive quiz. If people want to put some guesses into the chat box, that makes it even more fun. So what I'm going to have you do is guess what the fruit or vegetable is as we go slice by slice of MRI images through a fruit or vegetable. Give people maybe 15 to 30 seconds to get their guesses in. And just as FYI, some slides show multiple of a fruit or vegetable. For example, there could be 3 mushrooms in a slide. So if everyone's ready, I'm going to pull up the chat box, see if people are engaging with this. And if anyone's seen these before, I know I recognize a few names here. Maybe just withhold your answers. But yeah, here we go. Yeah. All right. So I see lots of rapid fire answers there. And indeed, this is a pineapple. So this picture here is not related to an MRI at all. This is just a picture I got off the internet. But the GIF here, this video, is from stitching together slice by slice an MRI image. Yep. Yeah. So a couple of right answers here. This is 3 cobs of corn, and we're just kind of going cross-sectionally through those cobs of corn. All right, next. Little trickier, quite beautiful, very geometric. Oh yeah. All right, we're converging. I see some right answers there. This is indeed an artichoke. And again, just going slice by slice through it. But we don't actually have to slice it up. The MRI does that itself. It can encode in 3 dimensions. We don't need to take a scalpel or a knife or a butcher's block at all to this. I think this is the last one. Ooh. Seeing people, no agreement so far. I see 1 correct answer, 1 almost correct answer. Okay, converging on cucumber. That's right. Or as I like to say, or I didn't realize that there was a few fairies trapped inside your cucumber, made out by all those seeds. All right, so back to some real work again. I'm just going to close the chat. So how do we actually get our MRI signal? Um, so here's an example looking at just a couple of, hydrogen nuclei, and it's showing their spin magnetic moment, and they're precessing in this kind of circular direction here. This is the spin vector. So the protons in a system will undergo microscopic motions, so they're moving about. They have translations, vibrations, rotations, collisions, uh, just like this. They're bouncing about, they're hitting each other all the time. And what's happening is, since each of these has a spin magnetic moment, they each are a little magnet themselves. So as a result, as they get closer to each other and farther away from each other over time, they're going to see kind of random fluctuations in the total magnetic field. So we're applying an external field with our solenoid superconducting coil, but each spin itself, each water molecule itself, is also going to alter the magnetic field very slightly. So this results in fluctuations of the frequency at which they're precessing, kind of randomly, and irreversibly then results in some dephasing. So if initially all those spins were in phase, As they see slightly different magnetic fields, they're going to get out of phase with each other by processing at different rates. And so this results in signal loss that's irreversible. So if we look at our signal as a function of time, it tends to decay exponentially with a time constant. We call it T2, which is spin-spin relaxation because it's influence of one spin on the other, or transverse relaxation because our magnetization is not aligned along B0. It's actually perpendicular to B0 at this point, or sometimes just called T2 decay. And then more relevant for functional MRI, is the fact that quite often we have an inhomogeneous magnetic field. Try as hard as we can to get a perfectly homogeneous magnetic field. There's always going to be imperfections in our magnet. And furthermore, the magnetic susceptibility of tissues are variable. So some tissues are diamagnetic, some are paramagnetic. So as soon as you put a person in the MRI scanner, your homogeneous magnetic field kind of vanishes and you get some inhomogeneity. So as a result, a spin, say on the right, is going to be processing slightly faster than a spin here on the left of this imaging region. And so you have your signal dephase even more rapidly as a result. And we call this then the apparent transverse relaxation time. We call it T2 star. It's always going to be less than T2. So if we look at this diagram, our T2 star decay is faster, so we lose signal more rapidly over time. So an example of where we use this in brain imaging is regarding the imaging changes around blood vessels. So our brain requires a continuous supply of oxygen, and this is transported throughout the body and throughout the brain, bound to hemoglobin in our red blood cells. So oxygenated hemoglobin, so while it's binding oxygen, is diamagnetic, so it has a negative magnetic susceptibility, which has the symbol chi, which is the same as most tissues. So our blood, when it's oxygenated, has similar magnetic susceptibility as surrounding tissues. But when it releases that oxygen at the capillary bed, and then goes downstream into veins, deoxygenated hemoglobin becomes paramagnetic. So our chi, our magnetic susceptibility, is positive. So if we consider just looking at like a cross section through a blood vessel, and in this top example, we're looking at an artery where the oxygen saturation is, say, about 95%. In this case, the magnetic susceptibility of the blood within the vessel is about the same as the susceptibility of the surrounding tissue. And if we look at another example of venous oxygen, its saturation is roughly 60%. In this case, the extra deoxyhemoglobin makes our blood slightly paramagnetic relative to the surrounding tissue. So chi blood greater than chi tissue. And we know that when we have differences in magnetic susceptibility across boundaries, we're going to end up with spatially varying magnetic fields. So you can go ahead and solve Maxwell's equations in order to determine, say, how a cylinder that has a slightly different susceptibility offset to its surrounding area will alter the magnetic field. You get something like these strong dipole patterns that you see around the vein, and then you see a weaker dipole pattern around the artery. So orange areas are showing areas where the magnetic field will be increased relative to your main magnetic field, and blue areas are those that are going to be decreased relative to your main magnetic field. And we know from the previous slide that when we have inhomogeneities in our magnetic field, this results in dephasing of our magnetization more rapidly. And because this is directly related to the oxygenation of the blood, we call this the blood oxygenation level dependent effect, or BOLD effect. This was discovered around 1990. So how do we go from this BOLD effect to actually measuring changes in brain activity? So let's see now what happens in the brain during changing brain activity. And first, let's consider our vasculature at rest. So we have cerebral blood flow, or CBF, that's supplying oxygenated blood to the capillary bed here where the vessels are all branching out. Oxygen is then extracted at this level, at this point, and consumed. We call that consumption the metabolic rate, or the cerebral metabolic rate of oxygen consumption. So that's oxygen metabolism. Oxygen is being used to meet the energetic demands of the neurons and surrounding tissue. So as a result, the blood in our veins is slightly deoxygenated, so it has more deoxyhemoglobin. We know that deoxyhemoglobin is paramagnetic and it perturbs the magnetic field. So at baseline, when we're just at rest, our MRI signal is going to be intrinsically low, especially around veins. During stimulation, say we're doing a task, we're watching a show, which is sometimes our resting state. But let's say we weren't watching it and then we turn it on and we're engaging with the show, or we're doing something with our hands, for example. We have an increase in brain activity. Our oxygen consumption has a slight increase to meet these elevated energetic demands. Our blood flow, though, has a very large increase, and our blood volume has a moderate increase. And we say that the blood flow increase is much larger than the oxygen metabolism increase. All of what's going on here, our blood flow responding dynamically to changing brain activity, we refer to as neurovascular coupling. So the neurons can communicate directly to the vasculature to make sure that we're receiving enough nutrients to sustain these dynamically changing brain activity levels. So this increase in blood flow actually delivers a oversupply of oxygenated blood. So this flushes out some of the deoxyhemoglobin in our veins, resulting then in a smaller perturbation of our magnetic field. So our MRI signal actually increases relative to baseline. So when we have increasing brain activity, we get an increase in our MRI signal locally where that brain activity changed. And this is now the basis of the bold functional MRI signal. So what does a typical functional MRI experiment look like? We have our subject lying in the scanner, and they're engaging in some sort of task. So quite often, us physicists, when we're testing something, we'll do like a visual stimulus. Or if someone's a vision scientist, they'll do some sort of visual stimulus. They might do something else, though, like finger tapping or having you do some sort of memory task. But in this example, let's say we're just looking at a blank screen for 30 seconds. And then for 15 seconds, we'll be looking at this radial checkerboard pattern, and it's going to alternate. The black and white blocks are going to alternate at 8 hertz between first white, then black, then black. And this really stimulates the visual parts of our brain. Then it turns off after 15 seconds, and we'll repeat this over and over and over and see what areas of the brain are activating. So we rapidly acquire a series of images on the order of every second or so. We can get a new image of the brain. And then what we can do is we can look at our measured signal from a given brain area. So here looking in the visual brain areas, which is near the back of the brain. And we can see it changes with time. And if we have a model for how that signal should change, like this yellow curve here, this is what we might predict. We can then correlate our measured signal with our predicted signal. And then we can generate a brain activation map, which is not really a map of how much did the brain activate, whatever that means, however you define activation, but it's really a map of statistical activation. So what areas were statistically correlated with our prediction? And that's what a typical fMRI experiment consists of. And so there's many uses or users of functional MRI. By far, the biggest player is in neuroscience. So using functional MRI to non-invasively study organization of the brain. So for example, we can understand sensory processing like this visual task, but more interesting things like musical improvisation, a response to stress, like if I was really nervous at the beginning of this talk, what parts of my brain are activating? And that's actually what these activation maps are. A lot of it occurs in the amygdala, which is a kind of a fear response part of the brain. Neurosurgeons often use functional MRI clinically, especially in academic medical centers, so they can determine what is the focus of, say, an epileptic seizure and try to remove that area, the epileptic focus, using functional MRI to localize the epileptic focus. Conversely, in tumor resection, if a neurosurgeon wants to remove a brain tumor, but they want to spare certain functions, like say you want your patient to be able to walk, so you want to preserve their quality of life. They can map out those parts of the brain that are associated with the so-called kind of eloquent, brain functions, and make sure that they do not remove those brain areas when doing their tumor resection. We can use fMRI to study brain physiology, looking at cerebrovascular health. This is an extreme example where blood flow is completely occluded to half the brain, and in response to a specific type of stress task for the brain, only half of it activated. But we can study more subtle things, including more subtle cerebrovascular impairment, but also Alzheimer's and dementia, also just how our brain changes with aging, and even COVID in the last few years, people have been able to do studies of how COVID changes the function of our brain. Us physicists use MRI or functional MRI and develop techniques, try to understand better the spatial specificity, make sure we know where our signal is coming from and how that relates to the underlying vasculature or neurons in the area, and then developing new contrast mechanisms that might be more specific to particular areas of the brain, for example. And then also a holy grail in psychiatry is to be able to put a single subject, an individual or patient into an MRI and see kind of like large scale changes to the network of patterns of their brain activity. For example, in ADHD or schizophrenia or depression, and then be able to stratify this patient and give them a tailored treatment plan to help them recover. So I originally was working mostly on studying kind of brain physiology and fMRI physics. And now I work also in addition to Carleton, I work also at a mental health hospital. So branching out more into psychiatric applications of these methods that we develop. All right, so let's recap here. The bold MRI signal is sensitive to deoxyhemoglobin in blood. So that's primarily located in the veins and the capillaries. The energetic demands of neurons are dynamically met by the vasculature to help supply nutrients to the local tissue, the neurons. We have a large increase in cerebral blood flow, resulting in an overshoot then in oxygenated blood. And that's why we get an increase in our blood oxygenation level dependent or bold signal. Also want to pause at this moment here to highlight kind of the value then of having interdisciplinary science. So I mentioned earlier that the bold signal was discovered in 1990 by a brilliant physicist named Seiji Ogawa. He worked at Bell Labs, which was kind of like, the Meta or Apple of today. Sorry, not of today, but of the kind of the 80s, 70s and 80s. And he wrote in this paper where he discovered the bold effect that we expect this oxygenation sensitive contrast could be used to monitor regional oxygen usages in the brain. When some region in a brain is much more active than other regions, the active region could show darker lines in the image, where the signal intensity would get lower, is what he's saying, because of the increased level of deoxyhemoglobin resulting from higher oxygen consumption. So this logic is perfect. If you're a physicist, you're thinking, okay, brain activity goes up, metabolism goes up, so I'm producing more deoxyhemoglobin. But he didn't realize that there'd been kind of a whole slew of physiology studies that showed that blood flow increases way more than metabolism. And as a result, you actually have this oversupply of oxygenated blood. And that's why the bold signal goes up and not down. So he spent a few years actually looking for these signal decreases and couldn't find them. And in the meantime, other research labs that were more integrated in a hospital that were working in a more interdisciplinary field rather than Bell Labs, which is, like hardcore physics, they were able to catch up and also, put out these real kind of first functional MRI experiment studies. And there was like a few labs that all kind of published this at the same time. It was a really exciting time. But I think it kind of highlights the fact that, it really pays off to have these collaborators and to work in an interdisciplinary nature because everyone specializes in their own field, and a physicist doesn't necessarily know, physiology. And so that's why it's good to kind of speak with outside of your normal kind of circles. And then, so finally, just to summarize, bold fMRI can map brain activity non-invasively. It does not use ionizing radiation, and it does not require any injection of contrast agents. So it's just using intrinsic changes in the oxygenation of our blood that are occurring all the time. But one kind of shortcoming of functional MRI is that it's still an indirect measure of brain activity. It's a signal that's related to changes in oxygenation, and brain activity is quite far upstream from that. So this is where my own research interests come into play, looking at physiological MRI. And so let's look a bit into that. And so first, let's just kind of step back. Where am I? Who am I? So I run the Functional Neuroimaging Physics Lab at Carleton University. It's down here. We're kind of sandwiched between the Rideau Canal here, which is famous for ice skating in the winter, and the Rideau River, which kind of comes along the southern end here. And in addition to working at Carleton, I also have an appointment at the Royal Ottawa Mental Health Center. The University of Ottawa Institute of Mental Health Research is there. And that's where we do our experimental research on a research dedicated 3 Tesla PET MRI scanner. So it does multimodal imaging. And the main goal of my research is to make MRI more sensitive and specific to both neuronal activity and brain physiology. I do this through a combination of biophysical modeling, so trying to kind of model the MRI signals from kind of first principles and developing new acquisition strategies and doing this in a synergistic way that we can use our modeling to inform our acquisition strategies that we're going to employ. And then also take real measurements and plug those into our modeling to make sure that our models are using kind of the most current physiological information. Okay, so let's go back to the main problem. So the bold signal can tell us where neural activity is changing, but not by how much. There's just too much kind of underneath the bold signal going on. There's changes in hemodynamics, so blood flow, blood volume, as well as metabolic changes like oxygen metabolism. And the combination of these two changes confounds then our interpretation of the bold signal. We don't know how much any of those physiological factors is changing. And when we look in pathology, so stroke, tumors, dementia like Alzheimer's, multiple sclerosis, head impacts, almost any pathology you look at today, there have all been studies showing that they have some associated cerebrovascular or metabolic impairment. So it really even further confounds our interpretation of bold signal if we're going to try to use it clinically. So the strategy is we try to acquire different sort of images then that are more weighted towards physiological parameters of interest. So here's some example of generating images of blood flow, oxygen extraction fraction in the middle, and the metabolic rate of oxygen on the right. So we have a whole slew of different sequences to kind of tune our contrast to these or sensitize our contrast to the different physiology. There's just a whole zoo of acronyms. I'm not going to go into them. And then we can use biophysical models to then obtain real quantitative estimates of the actual underlying physiology of these that contribute to our images, our MRI images. So measuring blood flow, blood volume, oxygen consumption, and again, a whole zoo of acronyms that I'm not going to go into. So let's focus in on that biophysical modeling part, this last part here, to try to get quantitative estimates of our physiology. So let's build up how we can kind of understand the bold signal. So we have some increase in brain activity. This is simultaneously leading to increases in oxygen metabolism, as well as a large increase in cerebral blood flow and a moderate increase in cerebral blood volume. This is what we were calling neurovascular coupling, that the neurons interact directly with the vessels around them. This results in a decrease in the local deoxyhemoglobin content and a decrease in the distortions in the main magnetic field, B0, and a resulting increase then in our bold signal. So if we look at, say, just a short impulse of brain activity, we might see a bold signal change over time that goes up and then returns back to baseline, maybe a bit of an undershoot at the end. So this is time on the x-axis, relative bold signal on the y-axis. Maybe this is on the order of 1% signal change maximum. So if we do a bit of accounting here of what's going on with our deoxyhemoglobin, what's going on with our bold signal, when we have increased oxygen metabolism, that's going to increase the amount of deoxyhemoglobin we have. When we increase our blood flow, that's going to decrease the amount of deoxyhemoglobin we have because more oxygenated blood's coming in. And if we have just more blood volume, we're also going to have more deoxyhemoglobin coming in too. So the impact they have on the bold signal, oxygen metabolism changes will decrease the bold signal, blood flow changes will increase by a lot, and blood volume changes will decrease our bold signal by a bit. So we can say that our bold signal is proportional to blood flow, cerebral blood flow, and inversely proportional to cerebral blood volume and the metabolic rate of oxygen. All right, so we're starting to get somewhere, but this was just very hand wavy. So others in the past have come up with a more formal model to describe the relationship between our changes in the bold signal and those same physiological parameters. We call this the calibrated bold model. So you can see it means our relative bold signal change is proportional to 1 minus blood volume changes, blood flow changes to the power of 1 minus beta, and oxygen metabolism changes to the power of beta. And what's really powerful about this is this now allows us to kind of invert this equation and measure changes in metabolism. And the reason we want to do that is because oxygen metabolism is more directly related to neural activity and brain health than the bold signal alone. And so this is one way that we can go about assessing or estimating oxygen metabolism. But a challenge is that we have to measure quite a few things here. So we'd have to measure bold signal, we'd have to measure blood volume and the blood flow, and this M parameter. We'll forget that for now. Blood volume turns out it's really challenging to measure. Bold signal and blood flow are a bit easier. So a simplified model was put out that just says that blood, so I'll just take your attention down to the second bullet point, that blood volume is directly proportional to blood flow through this power law relationship, through this exponent alpha. So this means that just small changes in blood volume result in large changes in blood flow. Alpha is a small exponent. And then now this model, we just need blood flow, bold signal, this M value, and then we can measure oxygen metabolism. But the main problem with this is that this alpha parameter here assumes that our vasculature is in a steady state, that any blood volume changes that are occurring have kind of slowed down or stopped. And that's kind of a main assumption that's typically not valid. Our brain activity is constantly changing. And so if those vessels are dynamically changing as well, then we're really never going to be in steady state. So we're asking in our lab, how, is this still valid away from steady state? And so here, this is showing relative changes in cerebral blood volume versus relative changes in cerebral blood flow on the x-axis. And we received experimental data from a collaborator who measured blood volume, which is very challenging, and measured blood flow during a vascular challenge where subjects actually inhaled carbon dioxide, which causes the vessels to dilate. And so we see here during the initial rise period, when our bold signal is starting to go up, blood volume and blood flow are both increasing. Then we reach a steady state period where vessels kind of stop responding. And then we turn off the CO2, we go back to breathing normal air. And then this is our return to baseline. So blood volume and blood flow are both returning to baseline. And this purple curve is showing kind of this steady state power law relationship between blood volume and blood flow. And we can see that it does a really poor job of capturing all of these hemodynamic kind of changes. It's pretty good in the steady state where it was developed for, but nowhere near good enough if we're trying to measure oxygen metabolism changes away from that steady state. So my PhD student, Yasmin Shafai, she wanted to understand if there's other models that we could apply to predict blood volume from a measurement of blood flow. And ideally using models that are more realistic and kind of derived from, again, sort of more first principles biomechanics. And so she's using a model of the vasculature that's kind of analogous to electrical circuit theory, where you can see here we can model the vasculature as a combination of resistors and capacitors in parallel. We call this a Windkessel model. I won't explain the historical significance there. We didn't come up with that term. And the analogy here is that electric charge is related to blood volume, current is related to blood flow, voltage to blood pressure, resistance to vessel resistance, and capacitance to vessel compliance. So how stiff are the vessels and can they dilate very well? So now we actually have a lot more parameters to fit, like what are these resistances and capacitances? And if we wanted, we could do this for arteries, capillaries, veins, like different categories of vessels. We don't have to do that. But now this is a much more biomechanically informed than just this power law. So we wanted to test how does this model fit? And we also compared several models. We used, again, this data from our collaborator of measured cerebral blood volume and cerebral blood flow time series in response to a range of different tasks in healthy participants. Those tasks include visual stimulus in the scanner, finger tapping tasks in the scanner, as well as this carbon dioxide challenge, which just causes the vessels to dilate and change. So here's an example time series now, looking at cerebral blood volume that's either measured, which is the black time series, across our participants over time in response to a very short, just 20-second finger tapping task or 24-second finger tapping tasks. And we're comparing a whole bunch of models, how they predicted that change in blood volume. And the power law that we started with is this dashed red curve. You can see that it's peaking too early and it's not getting quite the amplitude of blood volume change that we'd need, and really just lagging or opposite of lagging, I guess it's kind of leading the actual the measured changes. Whereas our Windkessel model is, coming up much higher, closer to the measured blood volume changes, and no longer lagging to the same extent the measured data. And so this was just one example of the tasks we looked at. We did 10 different conditions. We compared 10 different sets of tasks and conditions. And in all cases, this Windkessel model performed as well or better than all of the other models we looked at. And in the end, this then should provide a much more accurate estimation of any transient changes in oxygen metabolism we want to measure. So we don't have to use this simplified power law anymore. We can use a slightly more complicated Bin-Kessel model, but still get now, equal or better estimates now of metabolism. Okay, so that's kind of more in-depth. Now I just want to quickly overview some of the different research projects in my lab. So in addition to physiological MRI, we also do high spatial resolution functional MRI kind of development, both from the experimental standpoint, so developing new sequences to image at finer and finer spatial resolution. So here, this is showing an overlay of underlying kind of fMRI images and the activation maps in orange at a spatial resolution of 0.6 by 0.6 by 0.6 millimeters. So each pixel there is 0.6 millimeters isotropic, which is incredibly low. Typically, people are imaging at about 2 to 3 millimeters, not 0.6 millimeters isotropic. To better understand, where the signal is really coming from, which components of the vasculature are contributing, especially at high spatial resolution. We do biophysical modeling, so representing, the large vessels that kind of like cover the surface of the brain, as well as the microvasculature deeper within the brain, and understand how they each contribute to the MRI signal. This fourth year honors project student of mine did this year, and he developed that in a Python toolbox that we've developed and published that we call Bold Swim Suite. So it helps us implement all the sort of modeling tools that we want to use. So other physiological imaging projects I'm working on are trying to understand the MRI signal from blood, understanding how the packing and shape of red blood cells can impact the MRI signal itself, which is important for understanding how oxygen is delivered by the blood itself. Certain blood disorders like sickle cell disease can dramatically change the shape and morphology of the blood cells. So we can use these tools now that my student Jarell O'Brien is developing to try to understand how different pathologies can impact signal directly from blood. And then combining this all, I also have a project developing kind of very high spatial resolution vessel imaging, try to estimate blood volume and flow through kind of the smallest vessels that we're able to visualize. And we have more work on this, but just showing you the snapshot. So a postdoc in my lab was working on this, and now my PhD student is taking over this project. Okay, so quickly in the last few minutes, just want to kind of give an overview now of my own career pathway and kind of options within medical physics. So here's my random walk through academia. It starts in Winnipeg, where I did 1 year of engineering at the University of Manitoba. At that point, decided I was much more interested in some of the deeper concepts that the physics program was trying to answer, like kind of theoretical physics and particle physics. I was really into that. And at the same time, sort of neuroscience, and found that engineering at that point, at least what was available to me, was not looking into that. So I transferred after a first year of university to Bachelor of Science in Physics at the University of Victoria. I also flip-flopped a whole bunch between what I was studying. I did joint major in biochemistry and physics, and then took a organic chemistry course and thought, I really don't, it's not for me. So then switched into a math and physics joint program and took my first pure math course and thought, doing proofs isn't for me either. So then just went straight physics and really enjoyed that. I also was part of the co-op program, which gave me a lot of like hands-on learning experience, which I found really invaluable. Did a couple co-ops in particle physics labs and a few in medical physics labs, primarily at the Cancer Agency, working in the field of radiotherapy. After that, I moved for 1 year, followed my girlfriend, now wife, to Halifax, where she was from, and worked as a medical physics research assistant before going into grad school. Then started grad school. I did my master's in medical physics and my PhD in biomedical engineering, both at McGill University. But there's a twist there. My supervisor moved to Calgary, and I actually moved with him during my PhD while staying a student at McGill just for simplicity. And it was a nice excuse to get to go back to Montreal, which is a fun city. So then after my PhD, I did a postdoc in radiology at Massachusetts General Hospital at Harvard Medical School, yeah, down in Boston. That was a great experience. And then got my current faculty position at Carleton, yeah, in Ottawa. So I also want to talk just a little bit about medical physics more broadly within and use kind of the Ottawa scene for context. So here in Ottawa, we have what's called the Ottawa Medical Physics Institute. It sort of oversees our graduate medical physics program and brings together a network of physicists from all across the city in a huge range of fields. And I think it's a nice example of the sorts of types of jobs people trained in medical physics can go into. So we offer both a master's and PhD. In medical physics, our PhD is accredited by CAMPEP, so it means you can then go into clinical medical physics work later on. We have several faculty members at Carleton who specialize in medical physics, but then we have a much broader range of adjunct professors from a huge range of labs. So NRC, so we have national research labs kind of at the NRC, Health Canada, and Natural Resources Canada, who all contribute to our graduate program. So NRC, we're doing precision radiation dose measurements or dosimetry. At Health Canada, we have students working on the biological effects of radiation, also in space. You know, as astronauts go to space, they're exposed to more radiation. Natural Resources Canada, we have projects working on tracking, being able to track kind of radioactive sources, like for example, in a terrorist situation, if you're able to track down a moving radioactive source. And then we have more kind of conventional medical physics positions or research, say at the Ottawa Heart Institute, doing cardiac imaging primarily, at the Ottawa Hospital, doing both medical imaging applications, nuclear medicine, but also at the Cancer Center there in radiotherapy. And then also at the Mental Health Center where I work, there's a couple of us also supervising students with more of a kind of mental health and neurological disorders focus. We have a bunch of members in OMPI that aren't supervising students, but are just part of the network, share their expertise, and we have about 25 graduate students. So as you've seen, I've bounced around the country a lot. I think Ottawa has definitely the biggest diversity of research fields and breadth then of research fields that you really don't get anywhere else in the city. It's very rare. A lot of that is because of this capital effect of having all of these national research labs, scattered throughout the city. It's really a unique situation and position. All right, so let's conclude. So with MRI, we're able to generate a wide range of image contrast used in medical imaging. So for brain imaging, musculoskeletal, cardiac, etc. But also in research like neuroscience and other applications like imaging fruits and veggies, just for fun. MR physics is an active, vibrant, and very interdisciplinary area of research. So, I'm very heavily involved in the physics, but also the neuroscience side of things. So for me, I like my research passion is extracting physiological information from the MRI signal itself to help improve our understanding of healthy and diseased brain function. And medical physics more broadly, including MRI physics, we're applying our knowledge of physics to urgent clinical needs. And this is a field that's really in high demand almost all the time, and it pays well. So it's a reason to get some good grades now and get a medical physics grad degree, and you'll be nicely set in the future. No guarantees, of course, but that's been the case for quite a while in the years that I've been in physics. All right, with that, I'd like to acknowledge my lab and colleagues who've contributed their work to this, as well as research support from a few different funding support centers. And of course, I'd like to thank you for your attention and thanks for the invitation again, Megan. So I'm happy to have more discussion now.